Okay, so we have talked about the innate immune system in the previous lecture, and we have also just looked at the adaptive immune system. So I thought it's timely to do a quick consolidation, how the two immune systems work together when we are infected by a particular pathogen, right? So this is like a summary of a sequence of events that will happen during an immune response. Okay, just a summary. So you'll find that a lot of details wouldn't be included here. So anyway, this is kind of extra. I didn't put this in the notes, right? So I hope this helped those of you who actually spend time going through the SLS. Thank you very much. I know some of your friends don't really do that. So now that you do spend that time, so I thought I can help you a bit more, by giving you this summary. So I have two slides on it, right? This is the first slide. I have done it as kind of like fill in the blank, right? So I maybe you can pause the video and give yourself some time to try to fill in the blanks first before I go on to actually review the answers. All right. Okay, so anyway, for this summary, right, a sequence of events in an immune response. So obviously, the first thing that the pathogens would encounter would be the innate immune system, because it's an early response, right? So there would be inflammation, there would be the cellular responses, right, from the neutrophils and the macrophages and so on, right? So details, you can go back to the notes and look at it. And during this period, right, when the innate immune system is working, right, the antigen presenting cells, which is part of the innate immune system, they would be working as well, right? So they would phagocytose the pathogens once they have detected them, digest the antigens into fragments, peptide fragments, and display these peptide fragments on their cell surface via the MHC class 2 molecules. So that's where the antigen presentation would trigger the activation of a naive helper T lymphocyte or helper T cell if it has a T cell receptor that is specific for this displayed antigen. So this naive helper T cell would then bind to the antigen presented on the MHC class 2 molecule, aided by the CD4 receptor, and become activated. And once activated, it will start to release cytokines. So a naive B cell, also with a receptor, a membrane-bound antibody, right, a B cell receptor that is specific for this antigen found on this pathogen, would also become activated, aided by the cytokines that were released, right, from the activated helper T cell. Okay, so the activated B cell would undergo clonal expansion. It proliferates to form many B cells of the same specificity. Most of these B cells would differentiate into plasma cells that secrete antibodies of the same specificity. Some would differentiate into memory B cells. Okay, so the plasma cells secrete antibodies. These antibodies have the same specificity, right? So they can bind to the same antigens on the same pathogen that is now invading the body to prevent infection, and this is known as neutralization, and also to aid in the removal, right, by the macrophages, the monocytes, and so on. So that is known as opsonization, right? And of course, there are other antibodies function as well, which I'm not looking at in this summary. Okay, so I go on to the next slide, right? You can pause if you want to take some time to fill in the blanks yourself. All right, so I shall just go on. The cytokines released by our activated helper T cells also activates the cytotoxic T cells to kill any of our own cells that have been infected by this pathogen, right? So we call how the cytotoxic T cells themselves recognize the infected cells, right, by the antigen presented on the infected cells via the MHC class 1 molecules. Okay, and these cytokines, right, also activate other immune cells, or not activate, enhance the activity of other immune cells like macrophages. So macrophages are innate cells, right? Cells of the innate immune system, they are always active, right? They don't need to be activated, but their activity can be increased by the cytokines. They become more efficient in phagocytosis and so on, right? So the removal of the pathogen would be enhanced, okay? So once the pathogens have been removed, right, by the antibodies produced by the beast plasma cells, by the macrophages that are now working even faster, and if any infected cells appear, then by the cytotoxic T cells as well. Right, so once the pathogen, once the infection has been cleared, then most of these immune cells actually would die, right, via apoptosis. However, right, you have some that are long-lasting. They are called the memory cells, and they remain to provide the immunologic memory against another encounter, right, of the same pathogen, another infection by the same pathogen. All right. So this is a summary of what we have talked about so far. There are more to discuss, more to learn, right? But so far, this is what we have talked about. So I hope this serves as a good consolidation for you. All right. Okay.